Hello everyone. In paper 6, that is Genetics, Evolution, Biostatistics and Paleontology, under that the first unit, Genetics, we have discussed introduction, what are the theories that comes under the genetics, and we have also discussed about monohybrid cross, dihybrid cross and Mendelism. So in this session, we are going to discuss about Mendel's law of inheritance and use of Drosophila in genetic studies and dihybrid crosses in Drosophila. So the topic that dihybrid cross in Drosophila will be discussing in chromosomal theory. So in this session, mainly we will concentrate on Mendel's law and the use of Drosophila. So coming to Mendel's law of inheritance. So the Mendel, he gave around important three laws. So under that, the first law is the law of dominance. So law of dominance, so already we have come across the terms, that is dominant and recessive. So dominant means which will express in the offsprings or in the next generation, but whereas the recessive will not express in the next generation. So this law of dominance explains in a heterozygote, one allele may conceal the presence of another. So heterozygote and homozygote. So you know the difference between homozygous means under the same condition, heterozygous means they are of different. So this principle, its statement about the genetic function, and it tells some alleles evidently control the phenotype even when they are present in single copy. So here in the law of dominance, so something wants to be expressed in the next generation, or it has to be transferred from one generation to another generation, it has to be the dominant character. Then only you can see the character from parents to offsprings. So that is what the Mendel's first law explains, that is law of dominance. So coming to second law, that is law of segregation. So here he explains that two alleles of gene, they segregate or separate and enter gametes singly. So here the half of the gametes carry one allele, half carry the another allele. So that is what it is called as haploid. So two gametes, they fuse to produce a zygote that contains two alleles, so it becomes a diploid. So if it contains only the half, then it is called as haploid, and if it contains two gametes, so then it will be a diploid. So this is how the segregation or separation takes place in Mendel's second law. Coming to third law, that is law of independent assortment. So here the alleles of gene that govern two different characters. So if they are having two different characters, they segregate independently during the formation of gametes. So this is due to independent assortment in meiosis. So these are the three important laws which are given by Mendel. So the first one is law of dominance, second one is law of segregation, and third one is the law of independent assortment. Very important for the examination point of view. So next we'll see how the genetics or what are the history of this genetics. So in classical period, the genetics was taking place around 1900s to 1940, and later slowly the work of the Mendel was started to develop, and it was mainly studied by different types of scientists. And later that era is the modern period, so that started around 1970s. Currently now the genetics has evolved very much, and now we are moving into the genomics, or inside the gene or inside the complete structure, how the mutation, how the aberrations takes place. So that era is called as genomics or the proteomics. So these are the scientists who worked on the different era of the genetics, coming from the Gregor Mendel and Correns, Hugo de Virus and Teschmark. So these are the very important noble people who worked in the era of the genetics. So that is how we have come across the Mendelism, and we have completed the three important laws. So later we'll move on to the important theories which are later we will be studying in detail, that is chromosomal theory. So chromosomal theory, here the chromosome number, as I have said, it is constant within the species, but it varies among the species. So that is how the chromosomal theory, which was later discovered by Sutton and Boveri, and it was nothing but the continuation of the Gregor Mendel. So now in this we are going to discuss about the use of Drosophila in genetic studies. So we all know that the black flies which sit on the fruits and all, so usually they are called as fruit flies, and the scientific name is Drosophila melanogaster. So already I have discussed why Gregor Mendel used the pea plant. So the same reasons can be given for the Drosophila, why they are used in the genetics. So as we have already discussed, the pea plant, it is shortly living animal, and it has got the many different types of variations, and it is annual plant, so that it will live only for 1 year, and it can be cultivated easily in the normal environmental conditions. So that is how the same conditions can be applied to the Drosophila. So we are just making a note why these Drosophila are used in many genetic labs, and why the Drosophilas are extensively used as a model organism. So very important terminology, why we call them as a model organisms. So the fruit fly, that is Drosophila melanogaster, we are using as a model organism for genetic investigations. So as it has many characters which will make an ideal organism for the study of the animal development and the behavior, and in also neurobiology and human genetic diseases, and in different conditions. So as I have already explained, why we are taking the following characters, as they are having very small life cycle and short lifespan, and they can be maintained easily in ideal laboratories during the room temperature, and also show the different types of variations when treated with the mutants. So the many different types of mutants you need to study in your practicals also, like the white coloration in eye, red coloration inside the eye, and again the body variation. Some are very much larger, like yellow body, sepia body, and you are having black, completely dark black regions, like black body regions. So there are different types of mutants species you need to study in your practicals. So that's about this session. So in this session we have discussed about the laws of inheritance, and why the Drosophila is used as the model organisms in the genetic studies. So this completes the first subunit, that is introduction and the laws of inheritance. So that's about this session. Thank you.